academic career of our beloved Dr. D.M. Vasudevan. Once again, welcome you all for the 6th Endowment Oration instituted in honor of 50 years of academic career of our beloved Dr. D.M. Vasudevan.
Professor Dr. D.M. Vasudevan is the founder director of Jubilee Center for Medical Research of Jubilee Mission Medical College, Thrissur, Kerala. His leadership accomplishment has significantly influenced the day-to-day activities of JCMR. Under his guidance, Jubilee Research is now a recognized center of DSIR and Kerala University of Health Sciences. This center has Institutional Ethical Committee and Institutional Animal Ethics Committee, both approved by Government of India. The research center has MOU with different institutes of national and international importance such as National Institute of Occupational Health Ahmedabad, NIST, a CSIR institute at Trivandrum, CRC Kozhikode under Central Ministry of Social Justice and Empowerment, Canterbury Christ Church University UK, and University of Santo Thomas, Philippines. The publication graph of Jubilee Center for Medical Research has improved steadily with more than 100 publications in 2018. More of funder grants are coming to our way, and the latest feather to his cap is obtaining the Income Tax 35(1)(2) exemption status to JCMR granted by Government of India. Dr. Vasudevan is a distinguished personality in scientific fraternity. After completing his MBBS, he received his MD in Biochemistry from AIIMS, New Delhi, and FRC Path from UK. He is an authority in allergy and immunology and has worked extensively on cancer. Dr. Vasudevan received the prestigious Dr. B.C. Roy Award for Eminent Medical Teacher instituted by the Medical Council of India in the year 1992. He has also received the Best Doctor Award in Medical Education from Kerala Government and M.M. Ghani Award of Calicut University in the year 1994 and 95 respectively. His textbook of biochemistry for medical students is being followed by Indian and foreign universities, of which Spanish and Russian editions are also available. He has also authored textbooks for dental and paramedical students. He has guided 29 students for their PhD work. There are more than 240 publications in highly acclaimed journals to his credit. After his retirement from government service, he actively served at Kasturba Medical College, both Manipal and Mangalore, and as founder principal at Sikkim Manipal University and Amrita Medical College, Kochi. He continues to share his expertise with Amrita and Agape Diagnostics, Kochi. Dr. Vasudevan was the founder director of Research Institute at RCC Trivandrum as well as Amala, Thrissur. His sustained and exceptionally high-impact leadership achievements demonstrates the effectiveness in advancing the JCMR's goals and image.
An endowment oration was instituted in honor of 50 years of his academic career in 2019. The first orator was Dr. M.V. Pillai from US. The second orator was Dr. Shyam Kotilil from US. The third orator was Dr. Gagandeep Kang from Vellore. The fourth orator was Dr. Sumesh Philip Thomas from Canada. And the fifth oration in 2023 was delivered by Dr. Valsraj Menon, who was from UK. Let's begin the program. First of all, let me request the dignitaries Dr. D.M. Vasudevan, Dr. Vinod Scaria, Reverend Father Rennie Mundangurian, Dr. Previlal Kutichira, Reverend Father Paul Chalisheri, Dr. Rosmi Jose, and Dr. P.R. Verghese to kindly come to the stage and take their seats. Let's begin the program with a prayer. I request everyone to stand up for the prayer. We have a prayer team.
ഹൃദയരാഗതന്ത്രിമീട്ടി സ്നേഹഗീതമേകിയും കർമ്മഭൂമി തളിരിടുന്ന വർണ്ണമേകിയും നമ്മിൽ വാഴുമാദിനാമം എന്നും വാഴ്ത്തിടാൻ ഞങ്ങൾ പാടും ഈ സ്വരങ്ങൾ കീർത്തനങ്ങളാകണേ. ചുവടുവയ്ക്കും ഈ പദങ്ങൾ നൃത്തലോലമാകണേ. കുഞ്ഞു വീടിനുൾത്തടങ്ങൾ സ്വർഗമാകണേ. അമ്മ നൽകുമുമ്മ പോലും അമൃതമാകണേ. പൂർണ്ണമീ ചരാചരങ്ങൾ ഗുരുവരങ്ങളാകണേ. ഹൃദയരാഗതന്ത്രിമീട്ടി.
Kindly be seated. Now I invite Dr. Pia Verghese, Research Coordinator, Jubilee Center for Medical Research, to welcome the gathering.
A very good morning to one and all. Respected dignitaries, guests, faculty, staff and students, and benefactors of Jubilee. It is an honor to me to welcome all of you to this academic festival that we are celebrating for the 6th year. Dr. D. Vasudevan is our soul. We celebrated 50 years of his academic career in 2019 and started this endowment oration from that year. All these 56 years, this legendary gentleman has been living with his students, fellow researchers, and academic counterparts. Jubilee Center for Medical Research is proud to organize the 6th discourse of this endowment oration today. All about Dr. Vasudevan is already shown in the video just played now. First of all, let me welcome our research director, Vasudevan sir, who made this day possible to this function. Welcome, sir, with most lovingly and respectfully. The orator of this function, Dr. Vinod Scaria, is one of the best speakers on genomics in our country. When we invited him around 6 months back, Dr. Vinod was a senior scientist at Institute of Genomics and Integrative Biology at Central Government Institute at New Delhi. Being a medical graduate, his interest in genomics and bioinformatics is beyond our imagination. Now he is a senior consultant at Vishwanath Cancer Care Foundation at Bangalore. Dr. Rosmi Jose, Associate Professor, Department of Microbiology of Jubilee, his junior at Calicut Medical College, will introduce him with more details. Today, Dr. Vinod will speak on personal genomes in precision medicine. Welcome Dr. Vinod Scaria to this program. Now I request, wait. Now I request Dr. P.C. Rajiv, Associate Professor, Department of Emergency Medicine, his senior at Calicut Medical College, to offer bouquet and mark the reunion of old friends. Our director, Father Renny Mundangurian, will formally inaugurate this function. Father is our strength. Welcome, Father, to this program. Dr. Praveen Lal Kutichara, the principal of Jubilee Mission Medical College and Research Institute, is presiding over this program. Dr. Praveen Lal is our motivator. Welcome, sir, to this function. I also welcome Father Paul Chalisheri, our assistant director. Father always lends a great support to all endeavors initiated by the research department. Welcome, Father. I welcome Dr. Rosmi, Associate Professor in Department of Microbiology. She will formally introduce the speaker. Welcome, ma'am. I am so happy to welcome the friends and batchmates of Dr. Vinod Scaria, who are working in different hospitals in Thrissur, Palakkad and neighboring districts, who took pain to reach here. A hearty welcome, dear guest. I also welcome all academic persons from neighboring institutions. I could see Dr. Shajasar, Dean of Kuhas, and members from Amrita, and senior people from different institutions. A hearty welcome to one and all. I am extending a warm welcome to our CEO, Dr. Benny Joseph, all assistant directors, our medical superintendent, Dr. Shibu Sikallivalapil, to this program. I am happy to welcome all heads of the Jubilee institutions, faculty, staff and students of Jubilee institutions to this program. A warm welcome to media persons to this program. A special welcome to all JCMR team members, my own team members, who is working hard for the smooth conduct of this program. Once again, I welcome you all, wishing you a great academic feat. Thank you.
Thank you, sir. Now I invite Dr. Praveen Lal Kutichara, Principal, Jubilee Mission Medical College and Research Institute, to deliver the presidential address.
Respected teacher, Dr. Vasudevan, Kozhikode Medical College alumni, Dr. Vinod, dear colleagues, friends, youngsters. Even before the first primary health center came up in India, AIMS was conceived. When that was started as a 5-nation project, the first aim was to set trends in medical education. The success of that is remaining before us, personified as Dr. Vasudevan. He set trends in medical education. Once a 2-ward radiotherapy department in Trivandrum and a small side room as for cancer research, that is the name at that time, has now grown into regional cancer center. The name nobody can ignore in the field of cancer research. Sir was the head of cancer research to start with. He is a creator, creates from the vacuum when nothing is there. Sikkim Manipal Medical College, Amrita Medical College, leaving on all that, my experience in Thrissur Medical College in the erstwhile Ramvarma Sanatorium with only tiled roofs, all buildings, one building stood apart when I joined there. It's a concrete building, that is the Department of Biochemistry, headed by Dr. Vasudevan, and it has got a unique structure, lab plus a research lab and small conference rooms. And I learned this is the backbone for any research center. Shaji sir may be remembering those things. It is updating that model. Research team was carved from Thrissur Medical College. And Shaji is there now, research dean of university. Vasudevan sir is human, with humane attachment to everybody, at the same time keeping a divine detachment from everything. Long years I have with him. Never I saw him in anger or frustration. Only a small smile. Great teachers, many I have seen. But great teachers with disciples like you, most of you assembled here, I saw rarely. Wherever there is a program in the name of Dr. Vasudevan, flights will have tickets specific for India from abroad to come to that function. People from different parts of the country, mostly heading the institutions of research, will be here. That is guaranteed. That is the relationship Sir maintains with the students, especially the research students. My best wishes to Sir, Madam sitting in front, Madam, and to the program. Thank you.
Thank you, sir. Light symbolizes knowledge, purity, and is the dispeller of darkness. Now I request Reverend Father Rennie Mundangurian, the Director, Jubilee Institution, to come forward and formally inaugurate the function by lighting the lamp. May I request the other dignitaries to also join in. Thank you all. Now I request our director for the inaugural address.
Dear respected Vasudevan sir, dear Dr. Vinod, Dr. Pravilal, Father Paul, Dr. Varghese, Rosmi Madam, dear guest, to Jubilee family, and my dear Jubilee family members. Today is a day of pride and joy for the Jubilee family. Two things are happening here. First is our Vasudevan sir is there in amidst us as a center of point of today's program. In 2000, in Catholic Church, the Pope that time declared a year of great Jubilee, 2000. And one of the theme of that great Jubilee 2000 was like that: Jesus Christ was, is, and always. It was the theme of that year. I remembered this sentence when I remembered Dr. Vasudevan. In the 73 years old history of Jubilee, we have 7 institutions: hospital, nursing school, medical college, nursing college, College of Allied Health Sciences, and the 6th child is research department, and 7th is Ayurveda hospital. And when we started the research department for about before 10 years, this is the 10th year, this sentence comes to my mind. Vasudevan sir was, is, and always. Thank you, sir. Being with the Jubilee, making the research department a great success and dynamic department. Today, I can say Jubilee Research Institute is one of the most dynamic institute in Kerala. There was one email last week, we have not proceeded. Jubilee Research is shortlisted in the top 10 research centers in Asia. There was one email. Research centers in Asia. There was one email. We are contacting them. We got this email and we are contacting them for the details, like the Jubilee Medical College, 8th best medical college in India and the best in Kerala, that we got for 2 weeks ago. And last week we got an email telling Jubilee Research is shortlisted among the top 10 research centers in Asia. We have contacted them. The soul, Dr. Varghese told us, Dr. Vasudevan is the soul of this department. Like Dr. Pravilal told, he's a humane. He's a breeze, slowly but reaching everywhere, soothing everybody and inspiring everybody. The motive of the research department when we started here was to imbibe the instinct or the interest to research in all our departments. It was the motive. In all other 6 departments, beginning from the nursing school till medical college, all institutions, the members of all institutions should have this interest to research. The knowledge should be revived. The philosophy of Socrates, you might have learned. We had to learn in our during formation years philosophy. He told that the knowledge is inside. We have to take it out, and that is the role or the responsibility of every teacher. And I can say, not only because of the B.C. Roy Award, but from my experience, Dr. Vasudevan is the best model for that. Thank you, sir, for this good model. He is extracting this interest from everybody in our research department and all institutions. Sometimes the management has to break the power of the dynamics of the research department. So powerful is our research department. Last year, we have a very dynamic advisory board. 2 of them are here. They evaluated and reviewed the research department, and they suggested not going behind all themes, but concentrate on 1 or 2 themes. It was the advice from our respected advisory board. And 2 themes was the genomics and bioinformatics. And this year they came together again, the advisory board, and reviewed and told us our research department is following these steps, and they congratulated. So under the leadership of Vasudevan, Dr. Vasudevan, Dr. Varghese, they told us they are part of advisory boards in different institutions. Advisory board is meeting, putting suggestions, and go away. Next year come, and they see often it is not implemented, but it is not happened in Jubilee. They are implementing. So Dr. Vasudevan is before us with a small bracing smile. Thank you for your presence in Jubilee, sir. And following the steps suggested by our advisory board, the research department is choosing the orators for each year. This is 6th year, starting from 2019, and every year we got a good orators. And this year, following these steps, they were searching and found a renowned speaker on genomics. It's the theme advisory board suggested, and it is Dr. Vinod Scaria. We're happy to have him amidst us. The detail will be presented by Rosmi Madam, the junior to Dr. Vinod. I have also gone through who Dr. Vinod is, and I can say a fitting orator for this occasion. Hearty welcome, sir, to Jubilee. Wishing all the success. We have invited on the selected audience who can understand the topic. So only the selected audience is here who can little bit walk with Dr. Vinod. I wish all the success and having wish something new insight in this genomics. With your permission, I declare this oration is inaugurated. Thank you very much.
Thank you, Father. Dr. Rosmi Jose, Associate Professor, Department of Microbiology, will introduce the speaker. Over to you, ma'am.
respected dignitaries on and off the dais, distinguished guests, dear colleagues and students. It's my honor to introduce today's distinguished speaker, Dr. Vinod Scaria. So he's a clinician turned computational biologist. Presently, is a senior consultant to Vishwanath Cancer Care Center, Bangalore, and also an adjunct professor of IIT Kanpur. His core scientific areas are genomics, genome informatics and computational biology, genomic surveillance, and viral genomics. He is regarded as one of the pioneers in the area of clinical genomics in India. He is a co-founder of the Genomics for Understanding Rare Disease India Alliance Network, and this initiative helped to uncover the genetic basis of many inherited diseases in India. He was also instrumental in setting up comprehensive fellowship programs in genomics for clinicians. He is also a member of the HUGO Pan-Asian Population Genomics Initiative Task Force, and he has been a part of various collaborative genomic projects. And about his early life, he was born in Tanzania. He did his schooling at Calicut, and he is an alumnus of Calicut Medical College. After MBBS, he pursued PhD in computational biology from University of Pune. And he started his research career by joining CSIR Institute of Genomics and Integrative Biology, New Delhi. And during MBBS life, he was used to be a big part of junior academic club activities, and he's a gold medalist in biochemistry, physiology in Calicut University. Dr. Vinod has also made significant contributions in the field of COVID-19 genetic epidemiology. He spearheaded the state-level genomic surveillance programs for SARS-CoV-2 in India. He pioneered research into genomic characterization of vaccine breakthrough infections and reinfections. He is the propounder of COVID-19 open research data and resources, and he is well quoted in national and international press for this work. He has received various awards and fellowships. He is the recipient of CSIR Young Scientist Award for Biological Sciences. He is an elected fellow of Royal Society of Biology and Royal Society of Public Health. His research has also contributed to a number of commercially viable technologies. These include one of the methods for fast and accurate diagnosis of mitochondrial genetic diseases. He has more than 200 publications in international journals. He is the author of 2 books, and he holds a patent for human microRNA targets in HIV genome and method of identification thereof. He is also a member of editorial boards of many reputed international journals. And this is the testimony given by my batchmate. So we are proud juniors of Dr. Vinod Scaria, and I am extremely delighted to invite Dr. Vinod Scaria to deliver this prestigious oration on personal genomes for precision medicine. Thank you.
Thank you, ma'am. Now it's time for the much awaited oration lecture by Dr. Vinod Scaria. Let's listen to him.
Hello. Thank you very much, and a very good morning to all of you. It's always a pleasure to be here, and of course, we are all students of Professor Vasudevan because we all learned biochemistry through what he wrote many years ago. While he has not taught us in person, we are all indebted to him for having written those wonderful textbooks of biochemistry, without which many of us would probably not have passed biochemistry in medical school. Well, in today's talk, it's always a pleasure to be here in Jubilee Medical Mission Hospital, and it's my first time here, and I'm quite impressed about the work that you have been doing over many years. For the next 40 odd minutes, I will discuss about genomics, largely taking through my career in genomics, and also touching upon where it is going forward and how it's probably going to impact probably everybody on this planet Earth. Well, all that I'm going to talk about is work done by a fantastic set of people, colleagues, many of whom were students in my lab and have moved out in their careers, and also contributed by a fantastic set of clinical collaborators, around 300 clinical collaborators from across the country and across the world, and consistent funding from CSIR, which made this all possible. And all the work that I'm going to talk about has been from my former institute, the CSI Institute of Genomics and Integrative Biology. Now, before I start my talk, I think it is important to also take a side step and discuss something very parallel or very similar that has happened in modern medicine. Now, I'm not sure many of you would appreciate what this instrument is, but nevertheless, this instrument was created way back in 1971 by Godfrey Hounsfield, who eventually became knighted. Now, he was not a medical doctor, of course, it's not doctor, but he was a physicist to start with. He worked in the musical industry, making musical replicas, or what we call as discs. Now, what he did was even more remarkable, that he created this very interesting contraption, and this contraption could mean that you could put in human beings, a head of a human being for that matter. You cannot fit a head of a human being in this machine, but you can probably put in a mouse inside this, or a monkey inside this, and for the first time you can actually see inside of the human body without having to open it up, right? And I'm not sure how many of you would appreciate this fact that before this contraption was invented, if you were to look inside the human body, the only option was to open up the human body, right? And of course, people used to open up a human body after death, autopsy, and that's how anatomy textbooks were written over many years. But many more important fact is that you needed to open up a human body, a live human body, in case there is a pathology that was in there. Now, what I'm going to talk about is essentially a CT scanner, what we know today. And 50 years later, you would see CT scanners are all around. Right? You could just step out of this hospital and you will see at least 3 boards of a CT scanner being offered. Now, that's essentially what 50 years of a fantastic technology can really mean to medical care. And today, I'm sure no neurologist would pass exams in DM without being able to read a CT scan. Now, why did I bring this CT scanner in here? Because this is for the first time in human history that you could visualize inside of a human body without having to destroy the human body. And a similar technology today exists where you can visualize, or you can look at inside every human cells in terms of understanding the blueprint of what humans are without having to destroy the human body. And what I'm going to talk about is that we can today understand the genome sequence which is inside every one of your approximately 37 trillion cells without having to destroy your body. And this would necessarily mean that it would change the face of modern medicine as we practice today. Now, I was probably fortunate enough to be in the right place in the right time. I finished my medical school in 2003, and that was also the most important year for genomics, because a multi-nation initiative to sequence the draft human genome just concluded in 2003. And what this would mean is that you could now for the first time read the sequence of the human genome, which is approximately 3.3 billion alphabets from end to end, and in many ways nearly complete. And it took approximately $2.7 billion to do this initiative. And at that point in time in 2003, the discussion and the hope was that this fantastic technology would someday become commonplace in modern medicine. And that was essentially the hope and the outlook with which we left medical college in 2004. And I'm happy that we are on the right path to make this possible. Now, what happened really after 2003 in the last almost 2 decades has been something even more remarkable. While I said it took billions of dollars to sequence one human genome, the reference human genome, further genomes actually took much lesser, and the cost of sequencing nucleotides or sequencing human genomes is dwindling very, very rapidly over the last 2 decades. And what you see here is in a log order. It's not a linear scale, it's a log order. And that essentially means that the cost of sequencing is reducing at an unprecedented manner. We are already talking about $100 whole genome sequencing, and probably in a couple of decades time, it would be as cheap as a routine biochemical investigation. Now, what this would necessarily mean is that hundreds, if not tens of thousands, and possibly millions of people will get sequenced over the next decade or so. Now, if you really ask, is this sequencing happening in an equitable manner across the world? The answer is probably no, and this is a slide from way back in 2008. To make it very simple, what you have in the top panel of the chart is essentially the admixture plot of the world. It essentially means that how homogeneous or less admixed or more admixed are human populations. Each line is a particular individual. They're clustered by thicker lines which cluster them into populations. And essentially what you see here is the entire global population, right from Africa to the modern day Americas. And the bottom panel is essentially in 2008, the sequences of whole human genomes which were available. This included the reference genome, the Craig Venter and the Watson genome, which were the initial personal genomes to be sequenced, and then the Chinese, Japanese, and the Korean genomes. And what you see here is that they all came from much less admixed populations or largely homogeneous populations across the world. Now, the most admixed populations are in the middle of this chart, as you see here, from the Middle East to Central and Southeast Asia, which today encompasses almost pretty half of the human race on Earth, while we did not have any genomic representation. So that was the starting point on how we wanted to get into genomics and be able to sequence human genomes and understand this large population who seemingly were untouched by the modern day genomics. Now, India being one of the largest countries in this area and also in many ways diverse, there have been some work done in the past by my colleague Dr. Mithali Mukherjee, who for the first time used genetic markers to sort of understand how is Indian population organized. And what she found was that India is a very diverse country of around 4,000 odd endogamous communities, but nevertheless these endogamous communities could be largely clustered into 4 or 5 groups depending upon their genetic similarities or genetic affinities. But much more importantly, what she also showed is that India, being one of the hotspots for migration out of Africa into rest of Southeast Asia, being the larger landmass in that region, would also have representatives of all global populations around. Now, that necessarily means that India is probably one of the few countries which has practically all of world into one, but nevertheless clustered into thousands of communities or endogamous groups. These are groups of people who marry within the same community. By definition, this could be communities based on caste, or creed, or religion, and we still practice that today. But nevertheless, that necessarily means that the genetic material remains constant or concordant within those communities. Now, put this in the context of a large European concept of country. This would necessarily mean that India encompasses around 4,000 odd distinct countries. So in 2009, we were successful in sequencing the first genome in this country, which was announced in the parliament, and also becoming the 7th country with its ability to sequence a whole human sequence. We didn't really stop with one. We went ahead, collaborated, and extended our capability to the rest of many countries around this region, including sequencing the first Sri Lankan genome, contributing to the analysis of the first Malaysian genome, and contributing to a large number of programs in this region as part of the Human Genome Initiative called the PAPG. And much more recently, we ended up collaborating quite extensively with genome programs across this region, forming one of the largest databases for South Asian populations, and much more recently for the Middle Eastern populations. So the question is not whether we can sequence genomes anymore. The question is, how can we now quickly implement this understanding into clinical settings? So one of the problems that would require an urgent intervention is an area what we call as rare genetic diseases. Now we call them rare because individually these conditions are extremely rare. There are around 3,000 odd documented conditions today. Individually they might be rare, but put together these genetic diseases encompass around 70 million people in India. And this of course is an underestimate. Today we can estimate this to approximately around 100 million people. That would roughly mean that 1 in 10 individuals in this room would have a genetic condition, and many of you are not aware about it. Now the problem with people with genetic conditions is that they go through multiple physician visits, multiple rounds of misdiagnosis, and very few of them who eventually are able to get a diagnosis or a genetic workup. There's a considerable amount of time that is lost. And that would roughly mean that the point of intervention is delayed by around 7 to 10 years, and that is a lost opportunity. Because a significant number of them today are treatable, and if you can diagnose them very early on, you can institute a very routine treatment and they can have a normal life. So how do we really target this? Because if it is so prevalent as 1 in 10 in this room, but if you don't know that you have a genetic condition, how do you really find it? And the obvious question is, of course, clinicians might know how to find it, but nevertheless, that is not really possible. Now, the real other question is, for many of the scientific projects that we run, we really ask this question, is this advantageous to do it in India? And of course, genomics is advantageous to be done in India because we are a very large country. Today, 1/6 of humanity on earth is an Indian. That would necessarily mean that every rare disease ever documented in literature, we do have patients for it, if not hundreds of patients for it. And some of the disorders not yet documented in literature, we would have them in our communities. And unlike the rest of the world, since our population is diverse and stratified, we do have many more genetic diseases still remaining to be uncovered. And much more importantly, unlike the rest of the world, which has gone through 2 large world wars, 2 multiple rounds of famines and displacement, India largely has been undisplaced for probably 500 to 1,000 odd years, because practically everybody in this room can trace their ancestors by at least 3 generations. You all know where your grandparents are from, and that necessarily means that you can all draw a 3-generation pedigree. And everything in genomics starts, of course, with a pedigree, and since you can do that very often in India, unlike the rest of the world, also becomes a very good advantage to do this in India. Now, our foray into rare genetic diseases also was influenced by patients. And this is one patient who called up my colleague Dr. Sridhar, who has been a colleague and collaborator for the last 17 odd years, and and also a co-investigator for the first human genome. And since the genome was announced in December 2009, this patient saw this in news and reached out to Dr. Sridhar on phone and said, "What you did, I think, can change my life." And he introduced himself as Bhai. Of course, Bhai necessarily means that somebody of Gujarati ancestry. And what this individual had was a very peculiar genetic disease. As you see here, these are photographs of that individual. The skin would break up as if it was a bulla, and the skin was very fragile. So any point of friction or pressure would essentially break up like this, and it becomes infected and becomes a very painful condition. Now, today, of course, we do have a diagnosis for this, and of course, if you are a dermatologist in here, this is a condition called epidermolysis bullosa, a simplex variety, and not what you call a common genetic condition. It's an extremely rare genetic condition. Over the next few years, approximately 3 years, we personally have gone and met this family, and over multiple marriages drew an extended pedigree for this family, and that's in here. And all the red colored individuals are individuals affected with this particular condition. And there are approximately 28 individuals, if my count is right, affected with this rare genetic condition in an extended pedigree of around 5 generations. Now, what I'm trying to show here is, yes, there might be rare conditions in global literature, but the 28 individuals, one of the largest reported families for this disease anywhere in the world, across 5 generations, and we do have samples for every one of these individuals. The long story short, we could identify the genetic mutation in this particular family, confirming the diagnosis of epidermolysis bullosa simplex. So the mutation is in the keratin 5 gene, autosomal dominant condition, and therefore the hope to the family is that with the identity of the mutation, you can prevent the occurrence of this in the next one generation by appropriate genetic counseling and prenatal testing. So in many ways, many of these disorders which are thought as curses to families, science and genomics can essentially put an end to that curse, because we can understand the molecular basis of the disease, and with the right tools available already, we can prevent the disease from being transmitted to the next generation. Or in other words, we can stop this genetic disease forever. And many members of this family today have accessed genetic counseling, prenatal genetic testing, and I don't think there are any more family members in this family who have this genetic disease anymore. So in other words, one genome can change your life indeed, not just for yourself, but for an extended family who can get rid of the genetic disease from the community. So until 2015, we used to handle these cases on-off, but in 2015, with consistent funding from CSIR, we sort of put together a program which today is called Genomics for Understanding Rare Diseases India Alliance Network, or Guardian for short. Now what this program does is very simple. We work with clinicians in hospitals across the country. We used to work with around 70 hospitals, around 300 odd clinicians. And whenever a clinician sees a family with an undiagnosed genetic condition, they could refer us to the program. We would do a consenting, sampling, sequencing, and analysis. And whenever we arrive at a pathogenic genetic mutation, we could review it back to the clinician, and the clinician would counsel them or act upon them. And that happens in approximately around 50% of cases. The rest 50% of the cases, we need to go back to the lab, generate additional evidence to be able to prove that that mutation indeed caused the disease. And once we have adequate proof, we can actually go back to the clinician and be able to advise the clinician accordingly. And this largely helped close to around 10,000 odd patients in the last decade or so. I'll just give an example of one single case and how this program is similar or different from many other programs across the world, and how such programs could impact communities altogether. And this is after the program has been launched. This is one of the first cases that we handled, and this case came to us because the family had petitioned the President of India permission to euthanize 6 out of their 8 children. Just think of the plight of the family who would want to euthanize their own children. And that's largely because of the fact, as you see here, the family of 6 out there, they have a progressive neurological disorder from the youngest to the oldest in the clockwise direction. They're pretty normal to start with until around 3 years. And from around 3 years, as the history goes, they start going to preschool. They lose their milestones, eventually end up in a vegetative state, and eventually they die. And 6 children, one after another, without a diagnosis, without a hope. They went to the president, permission to euthanize all the 6 children. Now, of course, euthanasia of children, for that matter, even adults, is not permitted in the country yet. So, of course, this was referred to us through the official channels. We did a genetic analysis. We diagnosed this condition as cystic megalencephaly, or in many ways, in scientifically accurate terms, megalencephalic leukoencephalopathy with subcortical cyst type 1, an extremely rare genetic condition with a prevalence of approximately 1 in 3 million odd people. Now, incidentally, cystic megalencephaly is also a prominent genetic condition in a different community called the Bansal Agarwals, and there's a founder mutation called the Bansal Agarwal founder mutation, also known as the Wandernap mutation for this condition, also discovered in India. In fact, the 2 founder mutations for this disorder anyway discovered is in India. Now, for most of the clinicians and most of the researchers, the story ends here because the family has been helped, you have the diagnosis, you have probably the closure to this. Of course, this is not a treatable condition, a rare genetic condition in a family, 6 individuals, you found a new mutation, and probably that's the end of it. We publish the paper and forget it. But we were very curious, and that's what we made the difference, is where we went back to the community, and what we realized that they're part of a consanguineous group called the Nalbans. And Nalbans have a history because they seem to have migrated from a different part of the world. They seem to have migrated from the Mediterranean basin into India because the Nalbans used to make horseshoes. The alloys for horseshoes were very important in a period of time. So they came with one of the invaders. They formed the community called the Nalban. Today is approximately around 5 lakh odd individuals, and settled around Agra, Aligarh and Jaipur, but nevertheless have a common ancestry and a common founder. So we did a door-to-door survey of this community, sampled around 100 odd individuals within Agra and Aligarh, and what we found was even more remarkable. What we found is approximately 28% of community members were carriers for this extremely rare genetic mutation. And more importantly, we identified another 2 more families affected with the same genetic disorder and tested positive for the same mutation in a homozygous condition. Now, what would 28% carrier frequency mean for an autosomal recessive condition? Is approximately 1 in 400 children born to this community would have this genetic disease, which otherwise in the global context is 1 in a few million. Now, this necessarily means that while the rest of the world this disease is a rare genetic disease, for this community it is a public health problem, because 1 in 400 children born to this community would have this disease, and non-curable disease or a non-treatable disease. So that necessarily means that you need to provide this community with the right set of tools to be able to prevent this disease from recurring again in the community. And today that is possible with genetic counseling, with prenatal genetic testing and carrier screening. And to do this, you need to have a very affordable, cost-effective diagnostic available. And that's what we did in 2017 and 2018. By studying this entire community, we found this mutation. We converted it into very cost-effective assay based on Sanger sequencing, which would cost around 500 odd rupees. And this particular genetic mutation test can look at both the founder mutations discovered in India, both the Wandernap mutation, the Bansal Agarwal community, and the Nalbant mutation that we discovered. So the community members could be made aware. We work with community leaders and of course with Professor Shefali Gulati, who heads the pediatric neurology department. And enable through community education programs career screening of prospective couples who are at risk. They could do this test, do a career screening, and if they turn positive, they know the prior assumption what the child is going to have, they can do a prenatal test. In other words, you can provide the community with the right set of affordable, accessible tools to prevent the disease from recurring in communities. Now, in many ways, that necessarily means that you can take from the first case to a diagnostic today in the market. This diagnostics is today available in the Lalpath labs and many other catalogs, also called the Vandernapp and Nalband mutation screen. Community members can access not a whole genome sequencing, but a very affordable test which can solve their problem. Now, that will also open up a lot of more opportunities. This is not one such case, and many such cases that we have worked in individuals or communities which trace their ancestry from a different part of the world. Now, a lot of these people and communities, including in Kerala, their surnames, of course, that I've sort of mentioned, not an explosive list, today encompasses approximately around 15 million people who trace their ancestry from a different part of the world. They have all been Indians all throughout in culture, in tradition, in many ways of their lifestyle, but the genomics has a component which actually comes from a different ancestry. And many of them would also have very prevalent diseases which are prevalent in the Middle East, but now presenting in India, and therefore many of the clinicians are unable to diagnose them because they are not part of, in genomic context, the milieu that we see here. And therefore genomics becomes a transnational initiative, because what we understand is useful for others across the world, because there's an equal Indian diaspora, larger Indian diaspora of people of Indian ancestry who have moved to the rest of the world, and we also have people from the rest of the world who have moved into India for many generations now, and they have all been Indians in many aspects. So what we understand is beneficial to the rest of the world, and what the rest of the world understands is beneficial to treat our own people. So in many ways, genomics therefore is a brotherhood that we need to culture and nurture across the world. Now, while we all do this, we should also keep in mind that despite the best of our efforts, we might still not have a diagnosis. And this is one such case which was referred to us, I think, in 2016 rather, a child with a mitochondrial genetic disorder, which was referred to from the All India Institute of Medical Sciences. We did sequence the mitochondria, we got nothing. We did a whole exome sequencing, we got nothing. And therefore we were perplexed, and we ended up doing a whole genome sequencing. And that was when whole genome sequencing was becoming available at scale. And what we found was a very small deletion of around 100 odd kilobases, 100,000 bases in one of the child. And of course we sequenced the parents, and what we found is the parents also have a heterozygous genetic deletion, overlapping in a small region but non-overlapping for the rest of the regions. So the parents are non-consanguineous, they have very similar deletions, heterozygous, that means only in one of the two copies, but the child has a homozygous genetic deletion. Now, should we go back and tell this family that we have found this deletion and that causes the disease? Probably no, because this is a new genetic deletion. We don't know what it really causes. So we need to really go back to the lab to be able to understand what this genetic deletion does. And that's when you need to have model systems to be able to study genetic conditions. In this particular case, we made stem cells from the skin, fibroblast. We cultured them, did a mitochondrial assay to show that this indeed is a mitochondrial genetic defect. We made the same defect or a deletion in zebrafish, which is a vertebrate model system, which has pretty similar gene structure as ours. And what we found that the zebrafish also has a mitochondrial genetic deletion. So we have enough evidence to suggest that this deletion indeed causes a mitochondrial defect. And you could make a very cost-effective assay, because it's a deletion. You make an MLP assay, which makes it very cost-effective for the family. Now, of course, this could be done in a time scale of 9 months. That means that while the family could come back for the next pregnancy, you already have enough evidence to prove that the deletion caused the disease. You have a test to be able to do a prenatal testing for this family. And today, of course, after 3 attempts to conceive, the family could not, and the last 2 attempts were successful. They have 2 fantastic young children who are healthy, but nevertheless, end of a diagnostic odyssey for this family, from not being able to diagnose a condition to now having a novel mutation which runs in that extended community or an extended family. Now, there are many such examples, hundreds of such examples that I can quote, but of course for lack of time I will not do that. So over almost 7 years, we sort of built this collaborative network at scale, encompassing around 300 odd clinicians from around 70 odd centers, every major center in the country we used to work with. We converted it from a research program to an educational expedition where you could educate clinicians at scale. You could offer diagnosis for patients with rare genetic condition. And of course, you can generate evidence to be able to create policy. And today, fortunately, India has a national rare disease policy to take care of treatment of patients with genetic conditions. Now that opens up a very interesting question, right? I started with saying that these are rare genetic conditions, individually they are genetic conditions, but apparently if they are rare, that necessarily means that clinicians do not see it or clinicians do not diagnose it. Now, pertinent question is very simple. If 1 in 10 individuals in this room have a genetic condition, what is really prevalent? Right? If clinicians don't see it, I say that it is prevalent, doesn't make it prevalent. Now, this has been a problem, and it has been a global problem all throughout, because if you have any rare disease program committee, you call in 5 clinicians into the room, you call in a cardiologist will say cardiac channelopathy is the biggest problem. You call in a pediatric neurologist, he will say autism is the biggest problem. You call in adult neurologist, who would say muscular dystrophy is a bigger problem. So depending upon the speciality you call individuals from, you have a very different assessment of what is really prevalent in the community. Now, what really is prevalent in the community? Is there an answer for that? And the answer is nobody has an answer for that, because all these numbers essentially come from clinics, right? And clinics do have a bias, because if you know how to diagnose a condition better, right, you end up diagnosing that condition very often, right? And when you keep diagnosing that condition very often, a lot of your colleagues will also start sending you patients with that particular genetic diagnosis, and eventually that becomes a very prevalent genetic condition, right? The best example for that in Kerala is spinal muscular atrophy, right? Just because there is a lot of awareness, a lot of people tend to diagnose SMA, and therefore the apparent prevalence of SMA becomes extremely large. Now, that necessarily means that government will start acting upon it. The real question is, should we act upon evidence that comes from clinics, or do we have an unbiased way of estimating what is really prevalent? And to me, the only way to assess this is to be able to sequence apparently normal individuals, like you and me, who don't have a genetic condition. Let us sequence 100 odd individuals from this room and ask a very simple question: do you have a genetic mutation which can cause a genetic disease? You might be a carrier for the genetic disease, that's perfectly fine, but given your community, given your structure of the society, you can predict now what is your chance that you will marry another individual for the same genetic mutation, and therefore estimate the prevalence of a genetic condition. Now, that was not really possible, that was crazy, because there is no proof that this would work. But until 2019, we also were toying with this idea that we could probably sequence individuals in the community and be able to assess what is really prevalent in the community. And in 2019, fortunately, we got funding to sequence 1,000 odd genomes, and of course 1,000 odd genome is nothing compared to 1.4 billion people today that we live with. That means that one individual is approximately an approximation for 1 million odd people. So the idea was very simple. We will sequence 1,000 individuals, we will analyze their genomes, look at what is prevalent in the population, and now come back to educating clinicians that you are missing this diagnosis. And I'm sure I'm talking to a clinical community, right? There would be resistance, and you can never tell a clinician, a specialist, that you don't diagnose this condition. I know it is hurtful, but that is the reality. So we had this idea that we will go back and educate clinicians and now teach them a few tricks and give them a diagnostic test that they can diagnose patients very easily. I know this is very difficult, but we had some success in doing this, and this is from my alma mater, Government Medical College, Calicut. It took 5 years to convince my own professor, Dr. Geeta Govindaraj. that a certain set of genetic conditions were prevalent, undiagnosed, and was missed to the medical community. And these were patients suffering from primary immunodeficiency. That necessarily means that these patients do not have a patent immune system. They get an infection, they end up in sepsis, and they die. And infections in pediatric wards, I'm sure if the pediatrician is in here, is something very common, right? You see in, day out, patients who succumb to infections and sepsis, and you don't really try to follow them up on why did they get sepsis in the first place. But since from genomic information we knew that this is prevalent, we went back to create education awareness. We worked with the government to create a specialized pre-ID clinic and treatment for PIDs. And of course, the diagnostic support was offered by a philanthropic organization along with the state government. Now, this program has run for almost 6.5 years now, and we have diagnosed a consistent large number of patients with primary immunodeficiency disorders. Around 200 odd patients have been suspected and tested. A significant large number of people could access therapy, and the therapy is either give them immunoglobulins or give them a curative therapy, which is hematopoietic stem cell transplant, which today the government of Kerala supports. And many more families could access genetic screening and counseling so that they can do a risk assessment and they can also plan their future pregnancies. And given the prevalence of these conditions, there are around 400 odd genes in this particular condition, we are thinking of instituting a pilot newborn screening in Calicut Medical College to prove the real prevalence of the disease. Our estimate of the prevalence is approximately 1 in around 15,000 births. And in a medical college, Calicut, which takes around 30,000 births an annum, one of the largest in the world, that would mean that every year we could save approximately 2 to 2.5 all children, and we could extend this network across the state, and we would be able to save at least a dozen odd children every year. And these children can live a perfectly normal life with either immunoglobulins or hematopoietic stem cell transplant. So in other words, from genomic insights, you can indeed teach clinicians, of course, if they're willing to listen. You can institute programs very specific for prevalent diseases in that community, and more importantly, you can treat many of these conditions very effectively working together with the government. We have now done this over and over again. This is another such condition called familial adult onset myoclonic epilepsy. It's still not in the epilepsy classification. This is work done by Dr. Radha Mahadevan from Medical College in Tirunelveli. And she has been following up these patients over many years. Only in 2019 we did have a genetic diagnosis for this, and it's a pentanucleotide expansion disorder. There are only 13 cases reported in global literature for this condition, and Dr. Radha Mahadevan herself has around 400 patients from one single community affected with this genetic condition, and this is a treatable genetic condition. For such conditions, what we need is to have a cost-effective genetic test. We do have a cost-effective genetic test. We are still awaiting funding and philanthropy to support the initiative to screen the entire community, to identify people at risk and be able to treat them very efficiently. So there can be diseases which are extremely rare in global populations, but hundreds of individuals affected in Indian communities. Now, of course, when we talk about this, we talk about screening of individuals, and then the obvious question is, what should we really screen? And we talk about newborn screening programs, and I'm sure Kerala also runs a newborn screening program. And the choice and decision on what to include in the newborn screening program is largely divided upon what the company provides, right, and what the rest of the world does. Right? And typically there are countries who do 40 tests. Kerala, I think, does around 6 tests. But then the real question is, are we doing the testing for diseases which are prevalent in the community or diseases which are prevalent elsewhere in the world? But nevertheless, the basic concept of all newborn screening programs is that you can identify individuals or children at risk of developing a condition. Mostly these are biochemical tests. And if you can diagnose them very early on, you can put them to treatment very early on, and you can prevent the onset of the disease. So newborn screening programs are applicable for diseases which are diagnosable and which are treatable. Now the real question is, if that is the case, can we identify from genomic data what is the treatable, preventable causes in newborns? Right? So we ran through this 1,000 odd individuals, And what we identified was a set of disorders. Some of them do match with what newborn screening programs offer, like for example phenylketonuria. Not very prevalent in southern part of India, but now we know that it's very prevalent in the northern part of India. Carrier frequency is approximately 1 in around 300 odd individuals. But then of course there are disorders in the list which doesn't seem to be fitting into any newborn screening programs. Like for example Wilson's disease, carrier frequency is approximately 1 in 100 odd individuals. Cerebrotendinous xanthomatosis, carrier frequency is approximately around 150. But if you really look at literature, or if you ask clinicians, they don't seem to diagnose as much number of patients. But nevertheless, these are all treatable causes of intellectual disability. So we wanted to prove our point. So we went back. In fact, my colleague Dr. Binu Kumar went back to create a large cohort of individuals working for the last almost 5 years, working with clinicians with people suspected with Wilson's disease, trying to get them to a genetic diagnosis. And what today he has created over 5 years is one of the largest cohort of Wilson's disease in India. And what this would mean is that we do have founder events in the population. Some communities have very specific variants which recur in those communities. He also created one of the most comprehensive database for Wilson's disease genetic variants, reclassified all those variants, around 3,000 odd genetic variants. And I'm happy that Jubilee Medical Mission has been a collaborator on this. And of course created an AI methodology to be able to reclassify genetic variants, and also created a very cost-effective sequencing test which can be used for screening individuals. Now this would approximately mean that one in around 40,000 children born in India would have this genetic condition. There are, to put context, there are 25 million children born in India every year. And all these children, if they could be diagnosed very early on, they could be put on appropriate treatment. So many of these conditions are indeed real. At least for Wilson's disease, we know that they are real. Many of these patients are undiagnosed. Many of the patients are undiagnosed because Wilson's disease, if I'm sure pediatricians are in here, is a spotter in pediatric examinations. And that is a mistake that probably we are doing, because Wilson's disease, most of the patients might actually present much later on. And one of the under characteristic feature with which Wilson's disease appears is neuropsychiatric manifestations. And in fact, if you really go back to old literature on Wilson's disease, the first Wilson's disease patient indeed was diagnosed because he had a neuropsychiatric manifestation, was not a pediatric patient to start with. So there are some concepts that get ingrained into medical education by virtue of following the Western textbooks, or virtue of following certain patterns of recognition, but nevertheless that may not really reflect the real prevalence and phenotype of many of these genetic conditions. Now suppose that you can diagnose patients very effectively, and of course I'm sure you all treat patients day in, day out. And the other problem that we could address with genomics is to address a very common and known problem on why doesn't drug affect every individual in a similar way. And these numbers have been very well categorized now, and it is very well documented that the similar dose doesn't fit everybody on this planet. And this is an area where it is called pharmacogenomics. That necessarily means that there are genetic variations between me and you who are different, which could affect the way the drug is metabolized and excreted. And largely, to cut short, what we take in drugs is through the gastrointestinal tract, oral route. It's metabolized by the liver, which is called the first pass metabolism. It's distributed in the systemic circulation, and of course there are kidneys which excrete and metabolize in the urine, and the cells are in the extracellular space. These molecules need to cross over to the extracellular space, cross over the membranes, and reach the cognate target site. So in many ways they need to shuttle multiple barriers, multiple membranes, multiple metabolizing enzymes before they can reach the drug target site. And fortunately all of these components, what we talk about, whether carriers, transporters, metabolizing enzymes, are all genes encoded by your genome, and therefore genetic variations could affect each of this step, and therefore could affect the concentration of the drug which is available to the target site. And this is the basic concept of pharmacogenomics. And of course there is a lot of data on pharmacogenomics, and of course guidelines today available for a number of drugs. And of course what typically you could in the future able to do is, when every one of you would have your whole genome sequence, you would be able to look at all pharmacogenomic variants. You could go to the clinician's visit and show the pharmacogenomic report, so the clinician can actually decide that, oh, I'll not prescribe this drug and let me prescribe this drug, or should I change the dose of this drug, or should I give a larger dose for this particular drug. But I'm sure that's not really possible today. So you need to have very cost-effective ways of implementing this, and this is possible with genomic data because you can mine thousands of genomes, understand what is really prevalent in the population, and for what drug is that variation prevalent and effective, and therefore you can create very affordable, accessible pharmacogenomic test. So from the 1,000 odd genomes we could mine to understand what are the prevalent genetic variations. And for example, you can look at the frequencies of these genetic variations. Like for example, I'm not sure you appreciate, that's abacavir, an anti-HIV drug. The prevalence of adverse events, genetic adverse events, is around 4% in the population. That's so high that today if you give abacavir to a patient, it would be probably criminal to do that before doing the HLA test of the individual. Now, how do we really build such cost-effective genetic test? The answer is very simple. You can go back through genomes, mine through them, understand variations which are very frequent, revalidate in a small cohort of individuals, and be able to offer a very cost-effective, affordable test. And this is what we did with Amrita Institute many years ago. This is for an anti-cancer drug called 5-fluorouracil. The prevalence of 5-fluorouracil related toxicity in the Indian population is documented to be approximately 10 to 20%, one of the largest in the world. And therefore, we wanted to have a genetic test which can be preemptively used for patients who might get 5-fluorouracil. And therefore, we did this mining. We identified just 4 genetic variations which are effective in the Indian population. And therefore, for 4 genetic variations, we do have a test which today is implemented preemptively in Amrita Institute and many other centers across the country. So, mining genomic information can allow you to understand what is the prevalent genetic variations, and from the prevalent genetic variations be able to offer a very cost-effective, affordable test. They are not whole genomes anymore. They are RT-PCR tests to do now, and now you can preemptively do this test for individuals who are getting 5-fluorouracil. In fact, all solid tumor regimens today have one or the components of 5-fluorouracil or fluorouracils, including capecitabine and tegafor. And today, of course, this is available in many of the path lab catalogs. Now, having said that, I'll also set stage to what is going to happen in the future. Now, I said that cost of sequencing is reducing, and it is almost reduced to $100 odd in a few months to come by. And I think that is also a very interesting inflection point, because $100 roughly translates to the cost of doing a CT or an MRI, right? Which governments, individuals, and clinicians are very comfortable doing. Right? It's not an exotic, expensive test anymore. It becomes something that is amenable, affordable to a significant large number of people, and a significant number of people could now be able to access governmental programs because government also thinks that this is not a very expensive proposition. Now, while the cost comes down, that would necessarily mean that their probability of implementing that in routine clinical applications become very large, and more importantly, it comes consequently with the other graph that you see here is a number of genetic variations which would have clinical actionability and the number of sequences that is happening across the world. So today it's estimated that around 1 million odd people are being sequenced across the world as we speak. And to give a rough idea of what is going to happen, probably hundreds of millions of people are going to get sequenced in the next 1 decade. And probably in our own lifetimes, we would have every newborn child born with a genome sequence. And I think I can show you enough data to say that if there was an investment to be done on a child, a whole genome sequencing would be the right investment to be done. And that would also mean that many of the things that we practice in modern medicine today will undergo a drastic change. Right? What we do today is very simple in pediatric. If you're a pediatrician, or for that matter, any clinician, our medical system is designed and built on the concept that you can train doctors who can elicit signs and symptoms and a history, can arrive at a provisional diagnosis, do a set of investigations, confirm the diagnosis, and start treating the patient. Right? That is the concept of all that we teach medical students. But we have never asked this question. Thank you. We have never asked this question that if the patient already comes to the clinic, right, that means the disease has onset, and there's very little that you can do for once it happens. The real value of doing all of this is whether we can prevent patients from coming to clinics. Right? Does that mean that doctors will become jobless? Probably no. Yeah, so genomics today is part of the diagnostic workflow. The patient still has to come to the clinic with a set of signs and symptoms. The doctor makes a provisional diagnosis, and apart from a lot of biochemistry investigations, you do an exome sequencing or a genome sequencing to confirm the diagnosis, and so you say that, okay, you have Wilson's disease and therefore I will treat you. But the real opportunity in genomics is whether you can turn it around itself. Assume that a newborn was sequenced, right? You can identify that newborn would have sickle cell anemia or thalassemia, or that matter Wilson's disease, or that matter any disease that is treatable, and you can institute treatment based on evidence at the point of time. Right? And once you can do that, you can interpret and institute the treatment. That means that the patient is never going to develop that condition, right? And that really is, I think, the proactive way we need to turn medicine into, right? We should not expect that patients will come to you. We should expect that we will be able to treat the patient even before they develop their signs and symptoms, so that they can live a perfectly normal life, right? And that is the point of doing newborn screening programs, and probably that's the point in doing newborn sequencing programs, because newborn sequencing programs will cover all genetic disorders that are treatable. Today, even the best countries do around 40 genetic tests, mostly biochemical tests for genetic disorders. You can now improve that to hundreds of disorders which are treatable too. The second is, today we do a variety of genetic tests depending upon the case scenario. You might do a FISH or a karyotyping. I'm sure you would have a karyotyping facility in here. You would do a PCR based test. You would do an exome sequencing or a panel sequencing or a single gene test, so on and so forth. There are a variety of genetic tests that we do, including FISH and karyotyping. Now, each of the genetic tests looks at a kind of genetic variants, right? Nevertheless, these genetic variants are in the genome, but you look at a different kind of genetic variations. Each of these tests requires very specialized equipment, very specialized people who understand how to do that, and is also individually costly to do, right? And many often clinicians do multiple of these tests during the diagnostic workflow. Now, what I'm trying to do, all this test in one single test, and probably cheaper than any one of these tests, and more importantly, faster than any one of these tests. So in many ways, that would necessarily mean that all genetic tests eventually will make way to one single test, which is whole genome sequencing. And more importantly, their sequence is constant. The germline sequence is constant from the day you're born till the day you're dead. And that necessarily means that you can use this information throughout your lifetime, right? Without having to redo the test every now and often. You can use this test, or the clinician can use this test depending upon what stage of your life you are in, and depending upon the questions that you want to ask at that point in time. Just imagine that you want to prescribe carbamazepine. Are you going to do a genetic test at that point in time? Probably no. But if the patient already has the genome sequence, you can ask the genome sequence, do I have this particular genetic mutation? And if the genetic mutation is present, I don't give that particular drug. Or if I'm going to give abacavir, I'm not going to wait for 1 week to get an HLA test. I'm going to get the report of that particular variant then and there itself. So you can make real-time clinical decisions based on data that is already done. In other words, we would end up creating what we call as the next generation of digital diagnostics. The diagnostics would be done at birth. The patient will access that diagnostic report whenever the clinician requires it, right? With the advantage that every new diagnostic marker that gets incorporated during the lifetime of the individual will automatically become incorporated into the genome analysis, so that you don't need to redo this every now and often. So you have one test preferably done at the earliest possible when you're born, which you can use for the next 70, 80 odd years, depending upon what evidence the clinician would want to act on. And fourthly, and most important point, is that it is already faster to do a whole genome sequencing than any other genetic test today. You can actually sequence, analyze, and interpret and report a whole genome sequence of an individual today in less than 24 hours. There is no other genetic test today which you can do less than 24 hours. And more importantly, as a trend is you're seeing, it is going to be cheaper than any other genetic test. That would necessarily mean that whole genome sequencing would become one of the mainstays in diagnostic applications, and more importantly, it will move beyond diagnostic applications as newborn and early individual screening programs to identify genetic diseases which can be treated. And I think this is going to happen in the next 1 decade, at least for young individuals in this room. This is going to happen within our lifetimes, where we can see individuals who will step into clinics with a genome information and be able to use that information to aid clinical decision making. Now for that, there are 2 things that we need to take care of. One, we need to collect evidence at scale, and this is a problem in India very specifically, because as stated, we have 4,000 odd populations, groups of individuals, and therefore we need to systematically collaborate to collect information which is actionable for these populations. I'll give an example for one such initiative that we did. This is for BRCA1 and BRCA2, which are associated with hereditary breast and ovarian cancer. Carrier frequency in India approximately is around 1 in around 110 odd individuals and around 100 odd individuals. That means that probably 2 individuals in this room would have a BRCA1 BRCA2 mutation, would be susceptible to developing number of cancers apart from breast cancer, and the lifetime risk of developing breast cancer for the females is approximately around 80%. Many of you would have founder events which runs in the community or family, and the real quick question is that if you sequence individuals of BRCA and BRCA2, do we have appropriate evidence to be able to interpret upon? So what we did was very simple. We worked with clinicians and said that whoever has done a BRCA test any time, don't give us any other information other than the variant. We reclassified every single variant, created one of the most comprehensive databases today for BRCA, around 11,000 odd variants. But nevertheless, there's significant number of variants which still do not have a function assigned. And these run into thousands of variants. We cannot really do experiments at scale to be able to validate these variants. So what do you really do? You essentially go to looking at artificial intelligence. Since you already have a well characterized database, now machines can learn from this database to predict what is going to be the pathogenicity for a new variant when they pop up. And this today we can do it at scale, and what we show is that we can do it at very good accuracy, at least not for clinical interpretation and actionability, but at least for prioritization of variations that you can access for segregation studies or for that matter functional validation. So going forward, genomics largely is also going to be interpreted not by humans but by machines, but machines need to be created by humans. You understand the clinical aspect of it, the genetics aspect of it, and the interpretation aspect of it. And I think as clinicians going forward, at least the young clinicians who are in medical school, it is hard time that you learn genomics and genomic interpretation. And the other point that we need to sort of cover is how are we going to communicate this information in real life, right? Now, of course, you can sequence a whole human genome. That doesn't mean that your patient is going to come with a 1 terabyte hard drive and going to come with a doctor and say that please analyze this data. And that's not going to happen, right? At least in the world that we live in, right? You don't expect your clinician to connect that hard drive to a computer and now say that, oh, I look for your genetic variant, come back after 1 hour. We don't have 1 hour. So you need to have a system which can communicate this genetic information in real life based on what the clinician will require and based on evidence that is being created across the world. And the more important thing that we need to keep in mind is that while we can create data at scale, data doesn't really move from one place to another, right? To put numbers, there are millions of genomes sequenced while nobody is accessing this information because of one or the other restrictions, because people don't trust genomic information being shared widely, more so in India because we don't have a legal backing to do that. So we need to create systems both for evidence as well as for transacting this information in real life. And to do this, you can go back to other domains of regular life that we do, like for example, banking. Right? There was a time in our lifetimes, at least in my generation, we used to go to a bank counter, stand in front of that grumpy old man who would look at the ledger and say that, come after lunch. Right? Now, is that the way we want to transact genomic information? Probably no. Today, yes, we might be doing the same thing, but tomorrow we want to have a digital way of transacting this information where a clinician can swipe your card and be able to understand whether I should give you this drug or not give this drug. And we have one of the earliest pilots probably globally for enabling this. This was built in collaboration with a company called Zifo, and what essentially is called as an IndiGenome card. It's a secure, privacy protected, scalable, point of care and clinician enabled transaction system for genomic information. So it doesn't really transact your genome, it only transacts the evidence which is actionable at that point in time. Like pretty much like a credit or debit card, doesn't have any cash in it. But it enables you to transact at a point enough cash if required by an individual who is authenticated to do that. This was launched in 2019 by the Honorable Minister at that point of time. So in summary, before I close my talk, what we have built is essentially an ecosystem to start doing clinical genomics in India, working with clinicians and educating them, understanding the prevalence from population scale genomes, building rare disease programs to understand prevalence of many of these genetic conditions. By understanding them and understanding founder mutations, we could educate them and also offer very cost-effective, affordable genetic test. And by doing this over and over again, since the population at risk is very large, we could work with the diagnostic industry to enable this at much larger scale, and of course start using these data sets to build approaches for drug discovery in the future. In other words, what we have is a basis of, or the ways and means to do this at scale. And I think the next 1 decade, we need to do it at a much larger scale, because what we are currently doing is just scratching the surface of the problem. If we are talking about 100 million people in this country who have a genetic condition, we really need to think about a different scale approach to address this particular problem. And more importantly, what is really missing from all of this is education, right? And I'm sure as medical graduates, both you and even the youngest medical graduate would attest to the fact that genomics and genomic interpretation is not part of MBBS curriculum, right? And yes, of course, we can wait for the regulatory agencies to implement that, but we don't have time. This is going to happen in the next 1 decade, and we need to educate millions of doctors into understanding genomics. And the only way I think we can do that is to do a parallel route, to take it digital. And over the COVID time period, we do have very quite successful programs enrolled by around 4,000 odd participants from around 40 odd countries, making it one of the largest educational initiatives in the world. And we have also taken it further early to school students, because these are the students who are going to be 24 years in the next 1 decade. They're going to use genomic data in their routine applications, and they need to be aware today what they're going to see 10 years from now. And of course, we instituted one of the first fellowship programs in association with Sanofi. This is from a grant from Sanofi, which enabled training of clinicians in in-depth aspects of genomics. I think we need to do this more often, and more so in medical institutions like this, where we need to have education modalities at scale, where we can prepare the next generation of clinicians, and of course prepare a significant number of clinicians who are currently practicing to be able to understand and use genomics in their routine lives. Because if you don't do that, in the next 1 decade, when genomics becomes affordable and is becoming affordable, our population will miss out the benefits of genomics. I think I'll stop there and take questions, but before that, just a few summary points. Genomic sequencing is becoming affordable. It is not an exotic test anymore, and it will become part of routine clinical implementation. For an individual, it could mean a disease diagnosis. For a population, it could mean prevalence of genetic diseases, and therefore evidence for public health action. Genetic diseases are extremely prevalent in the population. Approximately 1 in 10 individuals, that means approximately around 20 individuals in this room have a genetic condition, and therefore genome sequences are for everyone. And more importantly, there is no time that is better to do a genome sequence. Of course, you can do a genome sequence for a newborn, but doesn't mean that old people should not do their whole genome sequences, because there is much more to learn from your genome sequence. Thank you very much.
Thank you, sir. Thank you very much. Since it's an oration, we'll have the discussion after the whole program. So thank you, sir.
Thank you.
Thank you for a wonderful and enlightening talk on genomics on Indian population. Now it's time for acknowledging our speaker. I would like to also inform the audience that the person associated with Dr. Vinod Scaria in groundbreaking research is Dr. Dilip K. Vijayan, scientist of JCMR. I request Paul Chalisheri, Assistant Director of Jubilee Mission Medical College, to hand over the memento to the speaker. I would also request Dr. Benny Joseph, our CEO, and Dr. Shibu, Medical Superintendent of Jubilee Mission Medical College, to join in. I would also request other dignitaries to kindly join the group. I would request Dr. Rosmi, ma'am, would also, if you could join. I would request Father Rennie, our director, to kindly join. I would also like to request our CEO to present the book, A Gentleman's Voyage to Dr. Vinod Scaria. This book delves into the life of Dr. D.M. Vasudevan, which was written by Dr. Kesia Kuruvilla, who was the Dr. Vasudevan's PhD scholar. Thank you, everyone. Thank you all. Thank you, everyone. Now I invite Dr. Matthew John, scientist, JCMR, to deliver the vote of thanks.
First and foremost, I would like to thank Almighty for making this sixth Dr. D.M. Vasudevan Endowment Oration a grand success. And my wholehearted thanks on behalf of entire Jubilee family to our speaker for today, Dr. Vinod Scaria, for giving us an enlightening talk, connecting to the heart and to the brain. കോവിഡിൻ്റെ സമയത്ത് ലോകം അന്ധകാരത്തിലായിരുന്നപ്പോഴത്തേക്ക് പ്രകാശം കൊണ്ട്, വിജ്ഞാനത്തിൻ്റെ പ്രകാശം കൊണ്ട് തിരിതെളിച്ച അപൂർവം ഭിഷഗ്വര ശാസ്ത്ര പ്രതിഭകളിൽ ഒരാളാണ് അദ്ദേഹം. അദ്ദേഹത്തിന് എല്ലാ ഭാവങ്ങളും നന്ദിയും ഞാൻ ജൂബിലിയുടെ പേരിൽ അറിയിക്കുന്നു. And next, my sincere thanks goes to our research director, Dr. D.M. Vasudevan sir, and his better half madam for coming over here and blessing this function, and your presence itself is a great honor for all of us. Thank you, sir. And next, my thanks goes to our team captain, our director, Reverend Father Reni Muttan-Kurian. He is always supportive to all our research activity. Thank you, Father, for coming here and blessing this function. And next, thanks to our dear Assistant Director, Reverend Father Paul Chalisheri. Achin is always very passionate about research and always asking us questions how this performs, how that performs, and that shows the tremendous interest that he has shown in research. Thank you, Father, for making this occasion. And next, my thanks goes to our principal, Dr. Praveen Lal. Sir is always supportive to all our research initiatives and activities and supporting us throughout our journey. And thank you, sir, for making this your presence for this function. And the master brain behind the entire concept, our dearest Dr. P.R. Varghese, sir. Sir is always supporting, and throughout he is very much involved in this program to make it as a success, and making sure everything is going rightly and perfectly, and as usual it always it happens like that also. And thank you, sir, for your presence. And next, my thanks goes to Dr. Vinod Scaria's colleagues who came over here and shown their love and affection towards their colleague, Dr. Rajiv and Dr. Rosmi. Thank you, madam and sir. And thank you for the medical superintendent, Dr. Shibu, for making your presence. Thank you, sir. And then the thanks goes to all the Jubilee administrative team, which covers the powerhouse canteen everywhere. They contributed their presence here to make this a success. And thank you, the entire team, Jubilee administrative team and management. And then to the faculty from all the departments who come over here with this great interest, and thanks from the bottom of the heart on behalf of JCMR for making this occasion a success. And your presence is very, very valuable to the success of this program. Thank you. And then to all the students, research scholars, and the residents, and the entire team. I don't know whether I have missed out anyone. Again, I forgot to convey my thanks to our beloved Dr. CEO, Dr. Benny sir, he is always supportive, and yesterday also sir was here, and I was just putting the tick, and just then I have seen the sir's presence is very, very auspicious. Your presence is a great motivation for all of us, and thank you, sir, for motivating us, making your presence here. And thanks to one and all again. Thank you.
Thank you, sir. Thank you all once again. And please join with us for a tea, which is served next room.